In the previous video, we have demonstrated how to access EdelweissData datasets through REST API in the JavaScript programming language. In this video, we will extend that example and show an interactive notebook for data analysis that heavily relies on EdelweissData's REST API service to support quick selection and analysis of published datasets. For this example, we obtained raw datasets of a PCR experiment from our Safer World by Design partner, BDS. The datasets were processed and published to EdelweissData following the steps demonstrated in our previous videos. The PCR experiments in this example were designed to answer two scientific questions. First is whether the metabolic enzymes are expressed in the cell lines under question. And the second is whether the expression of metabolic enzymes can be affected by typical chemical inducers. For this purpose, multiple cell lines were exposed to chemicals and gene expression profiles of metabolic enzymes were determined with PCR. Each dataset contains measurements for a single cell line. The end result of a PCR assay are normalized fold changes, which tell us how much is a particular gene expressed under given conditions compared to the untreated cell line. If the fold change is substantially greater than 1, then the gene is overexpressed under given conditions. And if it is substantially less than 1, the gene is underexpressed. The published datasets can be found on EdelweissData.com and then under the All Datasets section. The datasets are publicly available and their names start with a metabolic enzyme expression, like here. Beside that, we also compiled an overview dataset which lists the conditions of various experiments together with the links to their dataset. We will use this overview dataset to quickly link the selected conditions, that is the selected cell line, with the respective dataset that contains the process data for that cell line. This dataset was compiled once all the process datasets were published and each dataset got its unique ID in EdelweissData. Now I will switch to the Observable HQ notebook that I introduced briefly in our last video. This is a notebook based on the JavaScript programming language that has a very good support for interactivity. For example, it allows the user to select a certain option and then all the results are immediately refreshed. This notebook can be accessed through the link below the video. At the top, the notebook contains some general overview of this case study, description of the experiment, as well as explanation of data processing steps. Next, the user can select a single dataset or experiment from this set of experiments, and all the data is immediately refreshed. What happens in the background is that the notebook uses the EdelweissData's REST API service to access the data and metadata of selected dataset and displays it. As explained in previous videos, the metadata contains supporting information about the assay, like for example the additional information on the tested compounds. And since we are able to access this metadata in machine-readable JSON format, we can easily convert the compound data into human-readable format, like we did here. The table contains compound name, compound code used in this assay, and molecular formula of tested compounds. Below that, the actual data is displayed. First, we see a graph showing a single gene expression in all the tested compounds. Here we can select the gene as we wish and the graphs adapt accordingly. Further down, we see another graph, where gene expression levels are shown for all tested compounds and genes of metabolic enzymes as a heat map. Here, the color scale designates the gene expression level. The code that is used in this notebook can be found at the bottom and should be read from the bottom to top. You will see that we first import the required libraries, then we access the overview dataset, and once we pick a single assay, we use the overview dataset to interactively build a link to the dataset with PCR data and metadata. After that, there is basically just wrangling data around to achieve the desired format and display it. This notebook demonstrates how one can quickly access and visualize datasets published in EdelweissData through the REST API service that is offered by EdelweissData. Of course, the same thing could be achieved in other programming languages, but here we decided to use JavaScript because of its good support for interactivity, which makes it very convenient for such demonstrations. If you want to learn more about EdelweissData, please visit the edelweissdata.com.